Welcome to Sciencera. In this video, we're going to be discussing metabolism of carbohydrates. So this is chapter number 14 from Harper's Illustrated Biochemistry, Overview of Metabolism and the Provision of Metabolic Fuels. The learning objective for today's video is we're going to explain what is meant by anabolic and catabolic and amphibolic metabolic pathways. We're going to look at the outline of metabolisms of carbohydrates, lipids, amino acids, and at the level of tissue and organ and at the subcellular level. Then we're going to describe the ways in which flux of metabolite through metabolic pathway is regulated. Describe how a supply of metabolic fuels is provided in fed and fasting state. Let's begin. Metabolic pathways. The term metabolism refers to the interconversion of a chemical compound in the body as well as the pathway that individual molecules take. Their interrelationship and the mechanism that control the flow of metabolite via the pathways. There are three types of metabolic pathways. Number one, anabolic. Number two, catabolic, and amphibolic. Anabolic pathways are involved in synthesis of larger and more complex molecules from smaller precursors such as protein and triacylglycerols and glycogen stores. Endothermic anabolic pathways. Catabolic pathways are involved in the breakdown of larger molecules, often involving oxidation reaction. They are exothermic, reducing equivalent and primarily via the respiratory change ATP, and they are exothermic, reducing equivalents and primarily via the respiratory chain ATP. Amphibolic process. Such as citric acid cycles are found in metabolic crossroads and serve as the link between anabolic and catabolic pathways. Depending on physical activity, a 70 kg adult human requires roughly 8 to 12 mg or per day from metabolic fuels. Smaller animals requires more per kilogram of body weight while larger animals require less. To account for the energy cost of growth, growing children and animals have a corresponding higher needs. Carbohydrate 40 to 60%, lipids primarily triacylglycerols 30 to 40%, and proteins 10 to 15%, as well as alcohol are used to meet this energy requirement in humans. Depending on whether the patient is fed or fasting, as well as the duration and degree of physical labor, the mix of carbohydrate, lipid, and protein oxidized varies. If metabolic fuels intake exceed energy expenditure on a regular basis, the surplus is stored primarily as triacylglycerol in the adipose tissue, leading to obesity. If metabolic fuel intake is consistently lower than energy expenditure, there are negligible fats and carbohydrate reserve, and amino acid from the protein turnover are used for energy yielding metabolism rather than the replacement of protein synthesis, resulting in emaciation, wasting, and eventually death. When glycogen reserves are low, gluconeogenesis is fueled by amino acid derived from protein turnover. Insulin and glycogen regulate the production and utilization of triacylglycerols and glycogen stores, as well as the extent to which tissue take in and oxidize glucose. Diabetes mellitus. Diabetes mellitus is characterized by impaired insulin synthesis and secretion. Type 1 diabetes, also known as juvenile onset or insulin dependent diabetes, or impaired tissue sensitivity to insulin action, which is type 2 diabetes, also known as adult onset or non-insulin dependent diabetes, resulting in severe metabolic dysfunction. A table shows the outline of the pathways for the catabolisms of carbohydrates, protein, and fat. All these pathways lead to the production of acetyl coenzyme, which is oxidized in citric cycle, ultimately yielding ATP by the process of oxidative phosphorylation. So, carbohydrate, after digestion and absorption, become simple sugar, mainly glucose. Protein become amino acid and fat become fatty acids and glycerols. After their catabolism, they turn into acetyl coenzyme A, then goes into oxidize in the citric acid cycle, ultimately yielding the ATP by the process called oxidative phosphorylation. Carbohydrate metabolism and glucose. Glucose is the major fuel of most tissue. It is generated to pyruvate by the pathway of glycolysis. Aerobic tissue metabolizes pyruvate to acetyl coenzyme A, which can enter the citric acid cycle for complete oxidation to carbon dioxide and water, linked to the formation of ATP in the process of oxidative phosphorylation. Glycolysis can also occur anaerobically when the product is lactate. Glucose and its metabolite also take part in the other processes, for example, the synthesis of storage polymer glycogen in the skeletal muscle and liver, and the pentose phosphate pathway, an alternative to the part of the pathway of glycolysis. It is a source of reducing equivalent for the fatty acid synthesis and the source of ribose for nucleotide and nucleic acid synthesis. Triose phosphate intermediate in glycolysis give rise to glycerol moiety of triacyl glycerol. Pyruvate and intermediates of citric acid cycle provide the carbon skeleton for the synthesis of non-essential and dispensable amino acids, and acetyl coenzyme A is the precursor of fatty acid and the cholesterol, and hence of all the steroid hormones synthesized in the body. Gluconeogenesis is the process of synthesizing glucose from non-carbohydrate precursors such as lactate, amino acid, and glycerol. Lipid metabolism, which include fatty acid and cholesterol. Dietary lipid or de novo synthesis from acetyl coenzyme A generated from the carbohydrates or amino acids are the two sources of long chain fatty acids. Fatty acids can be oxidized to acetyl coenzyme A or esterified with glycerol to generate triacyl glycerol, which is the body's primary fuel source. Acetyl coenzyme produced by fatty acid oxidation can take one of three pathways. First one, it is oxidized to carbon dioxide and water via the citric acid cycle, just like acetyl coenzyme A produced by the glycolysis. Number two, it is a precursor of cholesterol and other steroid productions. Number three, it is converted to ketone bodies, acetoacetate and 3-hydroxybutyrate in the liver, which are significant source of energy during extended fasting and starvation. Figure on the screen shows the overview of fatty acid metabolism, showing the major pathways and the end product. Ketone bodies are acetoacetate, 3-hydroxybutyrate, and acetone, which is formed non-enzymically by decarboxylation of the acetoacetate. Next is amino acid metabolism, transamination. Amino acids are required for protein synthesis. Some must be supplied in the diet since they cannot be synthesized in the body. The remainder are non-essential or dispensable amino acids which are supplied in the diet but can also be formed from the metabolic intermediates by transamination using the amino group from the other amino acids. After deamination, amino nitrogen is excreted as urea, and the carbon skeleton that remain after transamination may be oxidized to the Carbon dioxide via the citric acid cycle be used to synthesize glucose from ketone bodies or acetyl coenzyme A, which may be oxidized or used for the synthesis of fatty acids. So in the figure you can see amino acid can form the non-protein nitrogen derivatives. Tissue protein can turn into amino acid, and amino acid can also turn into the tissue protein. When transamination occurs, amino acid turns into carbohydrate, amino nitrogen in glutamate, acetyl coenzyme A, and ketone bodies. Amino nitrogen in glutamate goes into deamination, forming NH3 and then urea. Amino acid metabolism. Amino acid resulting from the digestion of dietary protein and glucose resulting from the digestion of carbohydrate are absorbed by the hepatic portal vein. Liver has a role of regulating blood concentration of these water-soluble metabolites. In the case of glucose, this is achieved by taking a glucose in the excess immediate requirement and using it to synthesize glycogen or fatty acids. Between meals, the liver acts to maintain the blood glucose concentration by braking. Lipids are triacylglycerols that are hydrolyzed to monoacylglycerols and fatty acid in the gut, then re-esterified in the intestinal mucosa. Here they are packaged with protein and secreted into the lymphatic system and then into bloodstream as chylomicrons. The largest of the plasma lipoproteins, chylomicrons are also contain other lipid-soluble nutrients including vitamin A, D, E, and K. Unlike glucose and amino acid absorbed from the small intestine, chylomicron triacylglycerol is not taken up taken up directly by the liver. It is first metabolized by tissue that have lipoprotein lipase, which hydrolyzes the triacylglycerol, releasing fatty acids that are incorporated into tissue lipids or oxidized as fuel. Chylomicron remnants are cleared by the liver. Other major sources of long-chain fatty acid synthesis from carbohydrate in adipose tissue and the liver. The adipose tissue triacylglycerol is the main fuel reserve of the body. It is hydrolyzed in glycerol and non-esterified fatty acid are released into the circulation. Fatty acids are transported bound to serum albumin. They are taken up by the most tissue and either esterified to triacylglycerols for the storage or oxidized as a fuel. In the liver, newly synthesized triacylglycerol and triacylglycerol from the chylomicron remnant is secreted into the circulation in very low density lipoprotein. Partial oxidation of fatty acid in the liver leads to the ketone body production. Gly- Glycolysis occurs in cytosol and citric acid cycle occurs in the mitochondria. Compartment of pathways in separate subcellular compartments or organelles permit integration and regulation of metabolism. Not all pathways are equal to the importance in all cell. Depicts the subcellular compartmentation of metabolic pathways in a liver parenchymal cell. The central role of mitochondrion is immediately apparent since it acts as the focus of carbohydrate, lipid, and amino acid metabolism. It contains the enzymes of citric acid cycle, oxidation of fatty acids, and ketogenesis, as well as the respiratory chain and ATP synthase. Glycolysis, the pentose phosphate pathway, and fatty acid synthesis all occur in the cytosol. In gluconeogenesis, substrates such as lactate and pyruvate, which are formed in cytosol, enter the mitochondrion to yield oxaloacetate as a precursor for synthesis of glucose in cytosol. Membrane of endoplasmic reticulum contain the enzyme system for triacylglycerol synthesis, and the ribosomes are responsible for protein synthesis. Moving on to the flux of metabolites. To maintain an adequate supply of pathway product, it is critical to regulate the overall flux through it. It is accomplished by using regulatory enzyme to control one or more important processes in the route. The physiochemical parameters that regulate the rate of enzyme catalyzed reactions such as substrate concentration are crucial in control of metabolic pathways overall pace. Non-equilibrium reaction are the potential control points. Forward and reverse reaction occur at the same rate in either in an equilibrium reaction, therefore there is no net flux in either direction. In vivo, there is a net flow from left to right under steady state condition because substrate A is continuously supplied and product D is continuously removed. A metabolic route usually contains one or more non-equilibrium reaction in which reactants are present in concentration that are far from equilibrium. Enzymes that catalyze non-equilibrium reactions are frequently found at the low concentration and are exposed to the variety of conditions. The flux generating reaction is the first reaction in a pathway that is saturated with the substrate. Flux generated reaction is defined as a non-equilibrium reaction in which enzymes Km is significantly lower than the typical substrate concentration. Because its Km for glucose of 0.05 is significantly below the usual blood glucose concentration of 3 to 5, the initial reaction in glycolysis mediated by hexokinase is a flux generating phase. Rate of flux via the route is then controlled by the subsequent reaction. Allosteric and hormonal mechanism. The flux through this pathway can be regulated by the availability of substrate A. It also depends on the transport of substrate A into the cell. Flux is also determined by the removal of the product D and availability of co-substrate or cofactors. Enzyme catalyzing non-equilibrium reaction are often allosteric proteins subject to the rapid action of feedback or feed-forward controlled by the allosteric modifier in immediate response to the needs of cell. Frequently, the product of a biosynthetic pathway inhibits the enzyme catalyzing the first reaction in pathway. Metabolic fuels are interconvertible. Carbohydrate in excess of requirement for immediate energy yielding metabolism and formation of glycogen reserve in muscles and liver can readily be used for synthesis of fatty acid and hence triacylglycerol in both adipose tissue and liver when it is exported in very low density lipoprotein. The reaction of pyruvate dehydrogenase forming acetyl coenzyme A is irreversible, and for every 2 carbon unit from acetyl coenzyme A that enter the citric acid cycle, there is a loss of 2 carbon atom as carbon dioxide before the oxaloacetate is reformed. Fatty acid with an odd number of carbon atoms yielding propionyl coenzyme A as the product of final cycle of oxidation, and this can be a substrate for gluconeogenesis, as can the glycerol released by lipolysis of adipose tissue triacylglycerol reserves. Most of the amino acid in excess of requirement for protein synthesis arising from the diet or from tissue protein turnover yields pyruvate or 4 and 5 carbon intermediates of citric acid cycle. Pyruvate can be carboxylated to oxaloacetate, which is the primary substrate of gluconeogenesis, and the other intermediate of the cycle also result in a net increase in formation of oxaloacetate, which is then available for gluconeogenesis. Those amino acids that give rise to acetyl coenzyme A are referred to as ketogenic because in prolonged fasting and starvation, much of acetyl coenzyme A is used for synthesis of ketone bodies in the liver. Metabolic fuels is provided in both fed and fasting state. Glucose is always required by the central nervous system and erythrocytes. Erythrocytes lack mitochondria and hence always wholly reliant on the glycolysis and pentose phosphate pathway. The brain can metabolize ketone bodies to meet about 20% of its energy requirement. Remainder must be supplied by glucose. Metabolic changes that occur in the fasting state and starvation serve to preserve glucose and the body's limited glycogen reserves for use by the brain and red blood cells and to provide alternative metabolic fuels for other tissues. In pregnancy, the fetus requires a significant amount of glucose, as does the synthesis of lactose in lactation. Metabolic fuels is provided in both fed and fasting state. In fasting and starvation, glucose must be provided for the brain and red blood cells. In early fasting state, this is supplied by glycogen reserve in order to spare glucose. Muscles and other tissue do not take up glucose when insulin secretion is low. They utilize fatty acid and later ketone bodies as their preferred fuel. Adipose tissue release non-esterified fatty acid in fasting state. In prolonged fasting and starvation, these are used by liver for synthesis of ketone bodies, which are exported to provide the major fuel of muscles. Most amino acid arising from the diet or from tissue protein turnover can be used for gluconeogenesis, as can the glycerol from triacyl glycerol. Neither fatty acid arising from the diet or from the lipolysis of adipose tissue triacyl glycerols nor ketone bodies formed from the fatty acid in fasting state can provide substrate for the gluconeogenesis. Here you can see the summary of major metabolic features of the principal organ. Starting with the liver, major pathway for liver is glycolysis, gluconeogenesis, lipogenesis, beta oxidation, citric acid cycle, ketogenesis, lipoprotein metabolism, drug metabolism, synthesis of bile, salt, urea, uric acid, cholesterol, and plasma protein. Main substrate are non-esterified fatty acid, glucose in fed state, lactate, glycerol, fructose, amino acid, and alcohol. Major product exported by liver is glucose, glycerol, ketone bodies, urea, uric acid, bile salt, cholesterol, and plasma protein. Specialist enzymes are glucokinase, glucose 6-phosphate, glycerol kinase, phosphoenol pyruvate, carboxyl kinase, fructokinase, arginase, alcohol dehydrogenase. Next is brain. Major pathway for brain is glycolysis, citric acid cycle, amino acid metabolism, neurotransmitter synthesis. Main substrate are glucose, amino acid, ketone bodies in prolonged starvation. Major product exported by brain is lactate, end product of neurotransmitter metabolism. Specialist enzymes, those are the synthesis of catabolism of neurotransmitter. In heart, beta oxidation and citric acid cycles are the major pathway. Main substrates are ketone bodies, non-esterified fatty acid, lactate, chylomicron, and VLDL triacylglycerols, and some glucose. Specialist enzyme for heart are lipoprotein lipase, very active electron transport chain. In adipose tissue, lipogenesis, esterification of fatty acid and lipolysis in fasting are the major product. Main substrate for adipose tissue are glucose, chylomicron, and triacylglycerol. Major products are non-esterified fatty acid, and the specialist enzyme include lipoprotein lipase, hormone sensitive lipase, enzyme of pentose phosphate pathway. Then we have fast twitch muscles. Major product for this will be glycolysis. Main substrate glucose and glycogen. Major product exported are lactate, alanine and keto acid in fasting. Slow twitch muscles, major product is beta oxidation and citric acid cycle. Main substrate are ketone bodies, chylomicron and triacylglycerols. Specialist enzyme include lipoprotein lipase, very active electron transport chain. In kidney, gluconeogenesis is the major pathway. Main substrate are non-esterified fatty acid, lactate and glycerol and glucose. Major product exported by kidney is glucose. Specialist enzyme are glycerol kinase, phosphoenol pyruvate, carboxy kinase. For erythrocyte, which are red blood cells, major product for these are anaerobic glycolysis, pentose phosphate pathway. Main substrate are glucose. Major product exported is lactate, and the specialist enzyme include hemoglobin enzymes of pentose phosphate pathway. This is the summary of the major metabolic features of the principal organs. Clinical aspects. In prolonged starvation, as adipose tissue reserve are depleted, there is a very considerable increase in net rate of protein catabolism to provide amino acid not only as a substrate for gluconeogenesis but also as the main metabolic fuel of all tissues. In patients with cachexia, as a result of release of cytokinase in response to tumor and disease, there is an increase in rate of tissue protein catabolisms as well as considerably increased metabolic rates, so they are in the state of advanced starvation. High demand for glucose by fetus and for lactose synthesis in lactation can lead to ketosis. This may be seen as mild ketosis with hypoglycemia in human being. In lactating cattle and in ewes carrying a twin pregnancy, there may be very pronounced ketoacidosis and profound hypoglycemia. In poorly controlled type 1 diabetes mellitus, patients may become hyperglycemic both as a result of lack of insulin to stimulate uptake and utilization of glucose and because in absence of insulin to antagonize the action of glucagon, there is increased glycogenesis of further amino acid in liver. At the same time, lack of insulin to antagonize the actions of glucagon results in increased lipolysis in adipose tissue, and the resultant non-esterified fatty acids are the substrate for ketogenesis in liver. Utilization of the ketone bodies in muscles may be impaired because of the lack of oxaloacetate. In controlled diabetes, the ketosis may be severe enough to the result in pronounced acidosis. Acetoacetate and 3-hydroxybutyrate are relatively strong acids. Coma results from both the acidosis and also considerably increased osmolality of extracellular fluid, mainly as a result of hyperglycemia and diuresis resulting from the excretion of glucose and ketone bodies in the urine. This brings us to the end. Thank you for watching. If you have any questions, leave in the comment section. Do subscribe. Thank you.